Hello viewer, welcome to the YouTube channel FahadYounas. Today I will discuss the topic Meselson and Stahl experiment. The Meselson and Stahl experiment provided critical evidence supporting the semi-conservative model of DNA replication, the revolutionizing our understanding of genetic and molecular biology. Hypothesis. Meselson and Stahl hypothesized that DNA replication was semi-conservative, meaning each strand of the double helix serves as a temple for a new strand. Experimental procedure. Meselson and Stahl experiment provide that DNA replication is a semi-conservative process. They used heavy nitrogen to label the DNA and conducted centrifugation to separate the strands. Results and interpretation. Meselson and Stahl experiment provide that DNA replication is semi-conservative, where each new DNA molecule contains one original and one new strand. Significance and impact. Meselson and Stahl experiment confirmed the semi-conservative model of DNA replication, revolutionizing the field of molecular biology. The Meselson and Stahl experiment. The Meselson and Stahl experiment conducted in 1958 provided empirical evidence for the semi-conservative replication of DNA, which is fundamental concept in molecular biology. It was carried out by Matthew Meselson and Franklin Stahl at the California Institute of Technology. At that time, scientists were trying to understand how DNA replicates itself, and there were three proposed models for DNA replication: conservative, semi conservative, and dispersive. Conservative replication. In this model, the original double stranded DNA molecule remains intact and an entirely new double stranded molecule is synthesized. Semi-conservative replication. In this model, each new DNA molecule consists of new one strand from the original DNA molecule and one newly synthesized strand. Dispersive replication. In this model, the new DNA molecules would consist of short fragments of both original and newly synthesized DNA interspersed along each strand. To determine which of these models was correct, Meselson and Stahl designed an elegant experiment using the heavy isotope of nitrogen N15 and the lighter isotope N14. They grew E. coli bacteria in a medium containing N15 as the only nitrogen source, which resulted in all the bacterial DNA containing N15. They then transferred these bacteria to a medium containing N14 as the nitrogen source. Here's a step by step summary of the experiment. Bacteria were initially grown in a medium with N15, making their DNA heavy N15 DNA. After several generations of replication in the N14 medium, samples of DNA were collected at different times point. These DNA samples were then subjected to density gradient centrifugation, a technique that separates models based on their density. The N15 DNA being heavier salted lower in the gradient, while the N14 DNA settled higher. The key results of the Meselson and Stahl experiment were as follows. After one generation of replication in the N14 medium, the DNA settled as an intermediate band, indicating that the DNA was partially having N15 and partially light N14. After two generations, the DNA settled into two distinct bands, with one band at the intermediate position and one at the higher position N14. These results supported the semi-conservative model of DNA replication. In the first generation, the original heavy DNA had been split into two strands during replication, with each new strand pairing with a new light strand N14. As a result, the DNA became the intermediate density. In the second generation, the intermediate density DNA has replicated again, leading to mixture of heavy light and light DNA, confirming that DNA replication was semi-conservative in nature. The Meselson and Stahl experiment provided compelling evidence for Watson and Crick purpose mechanism of DNA replication, which has since become a cornerstone of molecular biology. Thank you for giving me the opportunity to present the Meselson and Stahl experiment. We appreciate your support for FahadYounas channel. Please consider sharing, liking and subscribing to our channel. Thank you for watching my video.